Hello students, welcome to E-PG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk about the anabolism of lipids. Anabolism of lipids from the paper Lipid Metabolism. If we look at the concept of the anabolism, it means that the preparatory phase of the lipids. And under this anabolism, the objectives of today's lecture is to understand what is anabolism, what are the implications of the lipid anabolism, and how the lipid anabolism is important and what are its significance as far as the life of an organism is concerned. If we date back to the history, we will find out that the word anabolism is derived from the Greek word. There are two words of Greek. One is called as ana, and ana means the upward, and bolism, and bolism means to throw. So there is an upward throw of the process, and this anabolism process have a propensity to build up the tissues, the organs, as well as a system. In this assembly of the process, the molecules, the small molecules, the small units, the small moieties, and the process engender differentiation and growth of the cells. That leads to the augmentation in the body size, the expansion in the body size. The reaction in anabolism entails and utilize energy that is released through catabolism, the exact opposite process of the anabolism, to synthesize the complex molecules which further employed to form cellular structures from simple and small precursors that act as building blocks. Numerous anabolic processes are power driven by the hydrolysis of adenosine triphosphate, which is also abbreviated as ATP, the storehouse of the cell, the powerhouse of the tissue. In the anabolism of the lipids, the precursor, that is the fatty acid, gets transformed into the triacylglycerol with the association of glycerol moiety. If we look at the biosynthetic metabolism process, or the mechanism by which the simple molecules get converted into a complex molecule. The external nutrients serve as the precursor of the simple molecules, and the sources may be from the intracellular or maybe intermediates from the linked metabolic pool of the synthesis of macromolecules. The conversion of a simple molecule or a monomer to a biopolymer takes place by the utilization of adenosine triphosphate, which results in the formation of the biopolymer. And this adenosine triphosphate serves as energy source for the anabolic process. As we can see here in the pictorial presentation of the anabolism of lipids, we can see simple molecules like ammonia, carbon dioxide, ethanol, lactic acid, urea, and many other monomer units gets transformed to complex molecule or the polymers like carbohydrate, proteins, or lipids. The reaction requires NADPH, where electron transfer takes place, resulted in the formation of NADP and hydrogen ion. As discussed before, the adenosine triphosphate serves as energy source for the reaction. In this pictorial presentation, the diagrammatic representation shows how enthalpy and entropy play an important role in anabolic as well as catabolic pathway. There are two types of reaction going on: an exergonic reaction where delta G is always less than 0, and endergonic reactions where delta G is always more than 0. The delta G represents the free energy, and in a cell, anabolic and pathway and catabolic pathway goes on simultaneously. In condition of low enthalpy and high entropy, catabolism takes place, and high enthalpy, low entropy also promotes the catabolic pathway. Cellular building blocks are formed, and during an endergonic reaction where low enthalpy, high entropy, and high enthalpy, low entropy, the anabolic reaction proceeds. Resulting in the formation of macromolecules. So energy is utilized during anabolic process, whereas heat is liberated during the catabolic process. If we look at the stages of anabolism, we will see there are different stages. The first and the foremost step of anabolism is the assembly of the precursor such as fatty acids, amino acids, isoprenoids, monosaccharides, as well as nucleotides. These are the precursor molecules which helps in the formation of the complex molecule or the building blocks required for energy generation. Here we are especially focusing on the fatty acid, which is a building block of lipids. When these assembly took place, it requires energy source, and that energy source comes from the adenosine triphosphate. So what happens? This activation of these predecessors or precursors into reactive forms happens when ATP provides energy source. Also, some of the cofactors like NAD, NADPH is also required to provide energy and to carry out the reaction. The congregation of these precursors into a complex molecules, for example lipids or polysaccharides or proteins or nucleic acid, takes place under different stages of anabolism. So coming to the anabolism of lipid, it is a vital component of metabolism. Lipids are essential and ought to be synthesized for numerous biological processes, and these numerous biological processes include the cell structure, the production of hormone, storage of excess calories. The nutrient metabolism, which is another important aspect of metabolism, is composed of both anabolic as well as catabolic reactions. The anabolic reaction blend molecules, whereas catabolic reactions break down molecules. A variety of the species of different organisms rely on diverse sources of energy. For example, autotrophs, especially plants, can make the complex organic molecules such as polysaccharides and proteins, or even lipids in cells from simpler molecules like carbon dioxide and water, using sunlight as an energy source. On the other hand, heterotrophs necessitate a source of more complex substances. For example, monosaccharides and amino acids to produce these building blocks. Likewise, phototrophs and photoheterotrophs acquire energy from light, while chemoautotrophs and chemoheterotrophs accomplish energy from the inorganic oxidation reactions. Fatty acids are a type of lipids composed of the chains of carbon atoms. The starting material for fatty acid synthesis is a 2-carbon molecule called acetyl-CoA that originates from carbohydrates, lipids, and protein metabolism. The formation of a fatty acid engages the repetitive sequence of 7 biochemical reactions that modify acetyl-CoA, allowing the covalent bond formation amid the carbons present in different acetyl-CoA molecules. Moreover, fatty acids are synthesized via fatty acid synthesis, abbreviately known as FAS, an enzyme complex, which polymerizes and subsequently lessen acetyl-CoA units. They include acyl chains that are unmitigated by a series of reactions that append the acetyl group and reduce it to an alcohol, desiccate it to an alkene group, and then diminish it again to an alkane group. The members of FAS complex comprise the condensing enzyme acyl carrier protein, a synthase, two reductase moieties, and a dry dehydratase. The enzyme complex is present throughout the body, especially active in the adipose tissue and liver. With each round of enzyme activity, the fatty acid molecule is amplified in size by 2 carbons. The product of fatty acid synthesis is palmitate, and palmitate is a 16 carbon molecule. In animals and fungi, all the FAS reactions are relayed by a single multifunctional type 1 protein. Whereas in plants and bacteria, a separate type 2 enzymes perform each step in the pathway. Other lipids like isoprenoids and terpenes outline the largest class of plant derived natural products. These compounds are prepared by the assemblage and variation of isoprene units. Contributed from the reactive precursors isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. In animals and archaea, the mevalonate pathway fabricates these compounds from 2-carbon acetyl-CoA units. In addition to its role in lipid biosynthesis, acetyl-CoA is central to the cellular energy metabolism. In this pictorial presentation, we are seeing the fatty acid synthase complex. There are numerous roles that fatty acid synthase performs, like self-renewal of neural stem cells, modulation of PPAR gamma in adipose tissue, calcium flux in cardiac and skeletal muscle, activation of PPAR alpha in liver, brain and macrophages, palmitoylation of endothelium nitric oxide synthase and MUC2 from the intestine. Another important anabolic process is the anabolism of triglycerides, or what we call as triacylglycerol anabolism. The building blocks, the fatty acids, they are the substrate for supplementary anabolic reactions to form triacylglycerols or triglycerides. In this process, the acyl transferases covalently bond 3 fatty acid molecules to a glycerol molecule to silhouette the formation of a triglyceride. So, from the association of 3 molecule of fatty acid and a molecule of glycerol in presence of covalent catalysis reaction by acyl transferases leads to the formation of the triglyceride molecule or the triacylglycerol. These triacylglycerols or triglycerides are the principal storage form of lipids in the body. The anabolic process of fatty acid and triglycerides inception are hormonally regulated, and these are regulated through insulin, and the progression of anabolic process is favored when the caloric intake exceeds the energy expenditure. The biosynthesis of triacylglycerol or the triacylglycerides is a supplementary anabolic reaction occurs by the catalysis of acyltransferases and the substrate were fatty acids. It is well recognized that fatty acid molecules, three molecules, linked with the glycerol moiety, solitary moiety of the glycerol, to form triglyceride. During the process, three water molecule gets released, resulting in the formation of triacylglycerol. However, if we look at the biosynthetic process of the triglycerides, it starts with acetyl-CoA. The acetyl-CoA moiety first gets converted to acetoacetyl-CoA and then to malonyl-CoA. This malonyl-CoA gets further converted to saturated fatty acyl-CoA through an enzyme desaturase 1 fatty acid synthase complex. And these saturated fatty acyl-CoA in turn regulate the activity of acetyl-CoA carboxylase. The saturated fatty acyl-CoA in presence of enzyme desaturase 1 gets transformed to monounsaturated fatty acyl-CoA, which further in the multi-step reactions pave the way for the synthesis of triglyceride. For example, the enzyme glycerol phosphate acetyltransferase helps in the transformation of monounsaturated fatty acyl-CoA to triglycerides. And these triglycerides itself, after synthesis, get stored in the body and utilized as soon as the requirement arises. So, students, today we have learned about the anabolic process of lipids, how the lipids synthesize, how the precursors, namely fatty acid, assemble to form the lipid molecule. For example, we have learned today about the assembly of the fatty acid 3 molecules with that of glycerol 1 molecule in presence of acyltransferases to form triacylglycerol or triglycerides. This is one of the simplest example of the anabolism of lipid. Likewise, other metabolic process, anabolism of other molecules including carbohydrates, lipids, nucleic acids took place in the body. And in the coming lectures, we'll continue with the detailed discussion of the anabolism of lipids in form of the biosynthesis of lipids, which we will be discussing in parts. Thank you.